Good morning, everyone. So you're all very welcome. So I'm just going to go through first what we'll cover today in the talk very quickly. So for the first half of the webinar, we'll be focusing very much on the more routine and regular testing that you might have to do and how to set that up. And I'll discuss why companies have to do that testing in the first place, and then talk about some of the issues they might face in setting it up. Then we'll have a look at where to find information on choosing the tests to do. So what tests to get done, the importance of sampling plans, and then ways to actually make good use of the data that comes out of all of that testing. And then in the latter part of the webinar, we'll move on to more specialized research type testing and have a look at how Teagasc can provide expert help in this area. And as Carol mentioned, of course, there'll be time for questions and answers at the end. Okay, so in terms of why we do testing, it's important to remember first and foremost that there's a legal obligation on all food businesses to ensure that the food they sell is safe to eat. So as I'm sure you've probably found to your horror, there's a whole raft of regulations around food production. And naturally enough, micro and shelf life testing is included in there. So as well as meeting your legal requirements, then testing data can provide you with a measure of confidence in the quality and safety of your product. For example, when you're trying to sell your goods, it can help potential customers decide if those goods are going to meet their specifications. So it can also be a great benefit then if you need to investigate food poisoning claims or defend yourself from any litigation arising from such incidents. And that does happen. I know a few years ago myself, I was asked to attend court in Cork because a client of ours was being sued over an alleged food poisoning case. And at that time, we were running an accredited testing lab in Teagasc. And because my name was on the report, I had to go to court. So on the day of the court case, the client's barrister presented the test results to the judge. And they were a really key part in having the case against them dismissed. The judge took on board all of their sampling procedures and records and the test results, and wasn't able to find any reason why the food served by them on that day could have caused an illness. You know, and in fact, the judge was quite annoyed that it ended up going to court because they had such a strong case. So it's really important then to have that sort of evidence behind you. Hopefully that will never happen to any of you. So one of the basic principles then of HACCP and other quality systems is to demonstrate that the controls you're putting in place are actually working. So where those controls are related to a microbiological hazard, the testing then will provide verification of the controls. So these last 2 points on the slide here, shelf life and product development, that they're very related in many ways, but we'll start with shelf life, I suppose. So determining your shelf life or confirming it is really important for the safety of your product and for the quality. It can also reduce food waste and might even give you a competitive edge if you have good shelf life. And when you're developing new products, you need to determine the shelf life of those products from the start. But then if you're reformulating them or maybe modifying your production techniques in any way, it's really important then to understand the effects that those changes might have on the growth of microorganisms in your product. So you do that by reassessing the shelf life. So these are probably the main reasons then for testing in my experience. So let's move on then to some of the issues that food businesses might face in doing that. So one of the most common problems that's noted to me over the years, especially by smaller businesses, is that they have no expertise on microbiology themselves or amongst their staff. So they might be experts in food production and they know their own products upside down and inside out, but microbiology is just like a foreign language to them. You know, and this is understandable, I suppose. There's such a huge range of tests you could possibly get done. It's hard to know which ones are right for your particular product or process. And as well as that, knowing what samples to take and when to take them and how to do it can also be difficult. And then even when they get their test results back, understanding what is written in the test reports can be hard for somebody who is not a microbiologist. We do tend to have our own terminology and we can be a bit fond of acronyms and other specialist jargon. So if I do slip into that, make sure you ask. If there's some term you don't understand, you can ask in the chat when we go through it later. So labs can and do have slight variations in how they report results and the types of units they use, and this can make it difficult for the food operator to decide how they should react to results. So they might know whether the results are good, bad, or indifferent or whatever. So we look at some sources of information here now to help you decide what tests you should be getting done. So I'm going to start kind of going clockwise around this image. So we're starting with your first stop, which should be the legislation. So this will guide you to what testing is mandatory. So you'll find the actual written laws in the Irish Statute Book website, but it might be easier just to go straight to the Food Safety Authority website, and that'll list the ones that are most relevant to food testing. So after the legislation then, there's the various guidelines produced by the Food Safety Authority. Although the Department of Ag and BIM also produce guidelines as well, specific to certain parts of the industry. So guidelines are technically not a legal or mandatory requirement, but they should be seen as a supplement to the legislation. And you need to remember though that this is what your environmental health officer or other inspection body will be working to. So in reality, if you're using best practice, then you should be able to meet the criteria contained in them. So some producers might find themselves then working with clients that set out specifications for the products they want to buy. So that'd be a written agreement between the two parties that outline the expected standard of quality for the goods, and that would generally include the microbiological quality as well. So this will give you another bit of information on what tests to look for. So another valuable source of direction in choosing tests is your own knowledge of your product. So don't underestimate it. So this might come from the work you've done already preparing your HACCP plans or risk assessments or your historical testing records. You know, and there are properties of your own product known as the intrinsic and extrinsic properties that you should become very familiar with, because these will guide you towards the types of bacteria that might be of concern for your products and ultimately towards the right test to select. So that's things like the pH and the water activity, packaging type, and so on. And Food Safety Authority guideline number 18 covers those in great detail. So it's really important to be familiar with that document, and I'll be reiterating that later on. And last, but definitely not least, you can always engage with external sources of knowledge. You could ask your private testing lab or your environmental health officer for guidance if need be, or you can engage with expert consultants like Teagasc to assist you in your test selection. Okay, so with regard to the testing that's set by law, the main piece of legislation, I suppose, for most food operators in relation to choosing tests is EC 2073. So this has been in Irish law for a number of years now, and it's updated from time to time. This year, I believe it was updated to include salmonella testing criteria for reptile meat, which sounds delicious. It sets out the criteria for a large range of food types, both at the point of sale as well as during processing. So that means your environmental or hygiene testing is included as well. It sets out when and where to do the testing and also gives possible corrective actions, which is helpful. It also lists when to use 2 or 3 class sampling plans. So a 2-class sampling plan, by the way, is where you have just a positive or negative result or a pass-fail. So there's only 2 outcomes. With a 3-class plan, your results could be categorized into 3 possible outcomes, such as satisfactory, borderline, or unacceptable. Okay, so it contains a lot of tables because there's a lot of different product types. So you have to search through to find the ones that are relevant to you. And it's really important to read all the small print after each of the tables as well, because they will outline exceptions and derogations and sampling methods and so on that might be important to you or your products. And finally, it's worth noting that that's not the only show in town, of course. There are pieces of industry-specific legislation in force, for example, in the areas of fisheries or dairy and water and so on. So as I mentioned earlier, most of the guidelines for the food industry emanate from the Food Safety Authority. And I don't think I can stress the importance of these two in particular. I still use these all the time to answer questions from my own work or for clients with queries around what organisms they should be concerned with. I still use it as a reference every day. So they're meant to be aimed at lay people as much as possible, but some of the concepts are a bit tricky, but it's worth trying hard to get to grips with them. So guidance note 18 in particular is the absolute must when it comes to setting or making decisions around the shelf life of your products. It sets out a number of ways to assess your products and the processes to guide you towards that all-important shelf life label. Guidance Note 3 then is more concerned with ready-to-eat foods, and it contains a number of tables setting out the criteria to be met. So it's a great guide on what testing to do. So remember, once you're producing foods, the Environmental Health Officer, the EHO, is entitled to take samples and send them off to the official food microlabs for testing. So therefore, if you know what they're going to test your food for, then you should be doing the same as a minimum. And just as an aside, the environmental health officers and the official labs have got together and published a handy list of all of the tests that they do for each type of food that they might take. It takes in all the criteria listings in Guidance Note 3, as well as EC 2073 and other sources. So it's quite a busy table, so I won't show it here, but I can circulate it later to anyone who's interested. Just let me know. So between legislation and guidelines, you should have a good idea of what tests might be required. So then what you really need to do is have a very good documented sampling plan in order to get the value out of your testing. So this is really important, yet it's something that a lot of companies seem to fall down on in my experience. So if you look at this diagram here, the items on the left is what guides what goes into your plan, and all the items in blue are the main things that I think you should have documented in your plan. So try and keep your plans clear and simple and specific to each product that you make if you're producing more than one. Your hygiene monitoring should be included in there as well. And it's important too to update the plan as you go along based on the results, or any changes in your products or processes. And once you've taken samples then, so it's important to label them well and get them to the lab as quickly as possible. So using the post is not really recommended for most sample types. It's best to use a courier or your own drivers. Some labs will collect them for you, or just drive them yourself if you have to. So this is another area where I've seen a lot of poor practice so many times over the years. When I was working in the accredited testing lab, we received literally hundreds of unidentified samples over the years, with no idea who sent them in or what testing they wanted. We've also had raw meat and other wet samples coming through the post. Believe me, on-post get really irate about that. And it's understandable. They don't particularly like handling post that's leaking blood or other unidentifiable liquids or solids. So as I said, some private accredited labs will collect the samples for you. And nowadays, many of those labs will have an online system where you can log in your samples before they even leave your premises, and that's really useful. So even if you do log them in advance, always put a letter in with your samples, given the sample details and the testing requirements. So if the lab doesn't have an online login system, then you should ideally email that letter in advance so that the lab can notify you if the samples don't show up. So when your results do come back then, always check them and react according to your plan. Don't just file the reports away and forget about them, especially if the results are not good. So before I move on, I'm just going to give a little example of where a company had no sampling plan and no reaction plan, and we were able to resolve an issue for them. So a number of years ago, a producer of meat products contacted the lab because salmonella had been detected in his product. So he hadn't tested his product for salmonella before, so he'd been advised by one of the regulatory bodies to set up a regular testing sampling plan. So that's why he came to us. We tested 2 more separate batches of his sample, and again, both of them are positive for salmonella. So he rang at that point then, and he was really concerned and stressed, and he was looking at maybe having to close his plant for a while until the problem was resolved. So we had a long chat together and to tease out his process, because I really wanted to know what way he made his product and how he controlled it and all that. And what transpired was that he actually had no sampling plan at all, and he had no action plan if there were problems. And it also transpired that he wasn't verifying the controls that he had in place, like he wasn't checking the temperature of his ovens. So we were able to help him set up a sampling plan, and then we put him in touch with somebody who could do temperature mapping on his ovens and make sure that they were operating to the correct temperature. And once all that was done, his problem went away, and he was able to keep his process in control for a long time. So had he had his plan in place in the first place, he would have saved himself a lot of heartache and stress for sure. So now I'm going to move on and say just a little bit about the test reports that you'll get back. So all of your good work is going to go to waste if the test report you get back from the lab is not clear and understandable. You're paying them good money to produce the report, so make sure that every part of it is clear and in a format that you can understand. So in terms of what's in the report, just check, they give each sample their own unique sample number when it arrives in the lab. So just make sure that their sample numbers tie in with yours and they've got all the details correct. Make sure that they've recorded when your samples actually arrived and what condition they were in and when they carried out the testing. You know, it's always worth knowing if your samples went on a free tour of Ireland in the back of a courier's van before getting to the lab, or if the lab themselves held on to the samples for a few days before testing. It's unlikely, but not unknown for that to happen. So when you get results back that are bacterial counts in the report, it's useful to make sure that you get actual numbers as well as log values or scientific notation. It's much easier to understand normal numbers, but log values might be required later if you're doing charts or trend analysis, and I'll talk a little bit more about that later. I don't really have time to get into the details of what log numbers are, but just to say that they act as a kind of a shorthand for when you're dealing with really big numbers, because bacteria can grow to really high numbers. Trying to create charts and graphs for such numbers is just not practical. So if you want to ask me about that later, you can. So you're required by the Food Safety Authority to use an accredited lab for your testing. So that's a quality requirement to ensure your samples are tested to the best possible standard, you know, in a well-managed lab. So the only thing to be aware with that is their scope of accreditation. Make sure that it covers your type of food as well as the tests involved. The labs themselves will generally just report results. They just give you that basic detail back. They don't tend to give you interpretations, but you can, and in my opinion, you should ask them to highlight any results that are outside the specification you want for those samples. So that's using a bold font or maybe a red font or something. So all you have to do is let them know your specification first. And they're usually very helpful and very, very professional about that sort of thing. Okay. So that result data that you get back then, that's the end product of your testing regime. It costs your business a fair bit of time and money and maybe even some heartache along the way. So it's important to use it as much as you can. Treat it like a really valuable asset. So your data should be stored away in a logical and ordered manner, but should be really easily accessible by you or your colleagues when required. Okay. So read the test reports carefully when they come in and react as quick as you can to any problems that might have come up. Okay, don't be tempted to file and forget. So even what you might consider to be a bad result is a valuable result. Those results would prompt you to take an action, and that might save the company money by preventing waste or recalls or even food poisoning incidents. Okay, so and having evidence of how you reacted to an out-of-spec result can be extremely valuable then for audits and inspections later on. Okay, so you can also use the data then to update charts, risk assessments, sampling plans, shelf life labeling, and so on. You know, feed it back into your quality system reviews and all that sort of stuff. So I'm going to talk a little bit about producing charts and graphs from the data, because I think it can be really useful, especially in the areas of hygiene and stuff like that. But or even anywhere where you're doing bacterial counts, it's always worth charting it to see how it's going. One of the main advantages of charts like this one on the screen is that you can often spot problems before they arise. And I've set this example up to highlight how that can work. So this chart here is based on the criteria for aerobic colony counts, and that's in guidance note number 3, table 6, if you want to look it up. So it's showing a 3-class plan where the bacterial count is considered satisfactory if it's less than 1,000 or log 3, borderline if it's between 1,000 and 100,000 or between log 3 and log 5, or unsatisfactory if it's greater than 100,000 or log 5, okay? So that band of green across the bottom of the charts is showing the satisfactory zone. The band of yellow color in the middle is the borderline zone, and the top red band indicates the fail zone. And for this example, the chart is showing results from a 14-day period. And the idea would be that you'd update a chart like this every few days as the results come in. Okay, so let's look at product 1 first. That's the black line on the chart here. So it's staying in the satisfactory zone for the whole 14 days. But the last 5 or 6 batches are showing it's kind of, there's an increasing trend. This is giving you a warning that there's something going wrong here and that the results are starting to creep up. And maybe the next ones could be in the borderline or even the unacceptable zone if you don't take an action. So that sort of trend, it would be really difficult to spot just looking at test reports because everything would look fine and you just put it away and you wouldn't react. Whereas this one will give you a clue that something is going wrong and you can react. So for product 2 there, that's the blue line. There are 2 batches in a row in the borderline zone. And guidance note 3 states that the food business operator should investigate the cause of this elevated level and take measures in order to return the levels to satisfactory levels for subsequent batches of food. So, you know, that's the same in all of these type of charts. As soon as you start seeing stuff in the borderline zone, you've got to take an action to bring it back down to satisfactory. So let's look at product 3 now, the orange line. There are 2 samples in the unacceptable zone, which are clearly out of norm compared to other results for this product. So as with borderline results, an action was required to prevent it occurring again. So in this case, though, the graph is showing there's a certain regularity to the poor results. Yeah, they're 7 days apart, day 5 and day 12. So it's possible that this might indicate that there's a problem related to a particular day of the week. So maybe it's a delivery day or a weekend day, or maybe there's a particular cleaning crew or operators on that day. So it would certainly get your attention and help you spot the pattern more clearly when you see it on a chart and therefore prompt you to take an appropriate action. So I hope I haven't spent too long on this point, but I do think it's really important to show just how valuable charts can be in terms of getting the big picture on your regular testing and using that to prevent issues arising. I have seen companies do this and they find it really, really useful. Okay, so to summarize a little bit before we move on to the more specialized testing that Teagasc can do for you. Just remind you to make use of all of the information sources mentioned to decide on your test and testing plan. Like, that's not an exhaustive list. I'm sure there are other sources around, but these are the main ones I would know. Document your goods and communicate the good sampling plan to all the relevant staff and get your samples in good condition to an accredited lab. Read the results you get back carefully, make sure you react to them, have a plan for reacting, and update the data then into reviews and plans, and make use of charts whenever you can to spot issues. And then just a couple of final points on testing. Testing without having something to judge against is, to my mind, a waste of money. I've seen people use this kind of a shotgun approach to testing where they test for every organism they can think of. And it rarely works. It's a bit pointless. Like, it costs a fortune to do that. So it's far better value to have a good think about selecting the testing to suit your product and processes. It's also a point of testing or effective testing that you can't test every single gram of food to check what's in it, because you'd have no product left. So what you're testing is really, really tiny amount of product compared to what you're actually producing. You know, it's kind of a snapshot of the overall picture at that particular point in time. And also it's worth remembering that bacteria are very unevenly distributed in most foods. So you can't necessarily guarantee that every other part of the lot or batch is exactly the same as the one you sampled. So that's where good reasoning and good thought comes into setting up your sampling plan, okay, to make sure that it's representative of the batch. So my final note then is that testing does take time, anywhere from 24 hours to 10 days. So it depends on the test. So just remember that for your plans and your distribution schedules. So we're going to look now at the more in-depth and less routine type of testing that you might require from time to time, and what we in Teagasc can offer you in that area. Okay. So sometimes you need a bit more background data to study the effects of a new ingredient or a process change, new packaging and so on your product. Or you might want to see if a particular pathogen is going to grow or survive in your product during its shelf life. So most of the private accredited testing labs don't undertake this type of work, or they don't have the facilities or the expertise for that. So to support the industry and type of work, Teagasc has built a new microbial safety and shelf life suite as part of the Prepared Consumer Food Centre. It's a bit of a mouthful, so I'm just going to call it the PCF lab from now on, if you don't mind. So the Food Safety Department is hosting that lab. While we've been doing this type of project work with clients for a long time, this is the first time we've had a really well kitted out, dedicated lab with all new equipment to help us provide a much better range of services. And the lab itself is self-contained, so we can safely work with the pathogens with no risk to our other food processing facilities that we have on site. So what can we do? We can work with you to undertake high quality research style projects designed specifically for your products and processes to help you determine the safe shelf life, or to determine effect of a product on the shelf life, or product or process change on that shelf life. So that can include challenging or spiking your foods with pathogens to determine if they grow or survive. And this is an area we have a lot of experience in through our own research activities, and also through work we've done with the accredited lab. And that's one of the things that really makes us very different from the private testing labs. We also have a range of processing, packaging, and storage facilities within the lab. So we can produce a range of different foods, and we can also pack them and store them under variable conditions designed to mimic what's going on in your own food chain conditions. We have some state-of-the-art genetic equipment that can, for instance, allow us to assess microbial viability in real time, or track changes in the variety of microorganisms in a food over time in great detail. Another feature of the facility we have, it's not on this graphic though, is that we can actually host microbiologists from your company to carry out work that you don't have the facilities to do in-house. So essentially you'd be renting the equipment and space, and you'd have access to the staff here. You just have your own person based here to carry out the work. Okay, so this is just a small selection of the more recent work we've undertaken to give you an idea of some of the variety of the projects we carry out. For example, there's a lot of interest in using natural antimicrobial agents to extend shelf life. So we do a lot of work in that area. So we're also doing a lot of research work in that area. So we have a good understanding of how that works. And we've also had quite a few projects based around reformulations, the effect that has on the shelf life of the product. So what I'll do next is I'll go through two examples of this type of project, slightly similar to each other, but they highlight some of the advantages we can offer, particularly around being able to spike samples with, or inoculate samples with pathogens. So I've had to change some of the details of the products and processes here in these examples to preserve confidentiality of the clients involved, but it won't affect what I'm demonstrating anyway. So in this first case here, the client wanted to challenge a raw meat product with Campylobacter and follow that with a combination of antimicrobial sprays and ultrasound treatment to see which combination of those would best kill or at least suppress the pathogen. So first and foremost, we undertook some pre-trials with the product to see what ultrasound parameters were going to be the most useful without changing the appearance or texture of the product. We didn't want it to end up looking cooked after the ultrasound. So starting at the bottom left of this diagram here, and moving clockwise, we inoculated the product with Campylobacter. Basically, we put Campylobacter into a liquid and dipped the meat into it, took it out again. We split it into lots and applied the different antimicrobials using a sprayer. We then vacuum packed the product and treated it with ultrasound in a water bath. And then we stored it in chill and tested it during storage. So the client at the end of that got a lot of data that they were not in a position to generate themselves, and they were able to then select the most effective combination of treatments for their own project. Okay, so in the second example then, the client was considering the use, again, an antimicrobial spray product, and he wanted to assess if it would be effective enough on his product to warrant the purchase of new spraying equipment for his plant. So to start it off, we did some initial work, and that's what's shown in this graph here. We wanted to make sure, first and foremost, that the antimicrobial would be effective in lab conditions. we grew some Salmonella strains in various concentrations of the antimicrobial in the nutrient broth. And so if we look at the pink line here, this is the growth of the Salmonella over time, just in the nutrient broth on its own. You can see it increases quite happily over time. So when we added the antimicrobial at 10%, there was a tiny bit of growth, but very, very little. And when we added at 30% strength and 50% strength, it completely flatlined the growth of the Salmonella. They weren't growing at all. So the results were very encouraging, and it indicated that the antimicrobial was highly effective against Salmonella in the lab, or in vitro at least. So we could then move on to the more real-life experiment with the meat. So this graphic is showing what we did in the second part of the trial. And again, we're moving clockwise here around this diagram. So first we inoculated the product with Salmonella again and applied the antimicrobial by spraying. You know, and we used the parameters that the client set for spraying, how much spray to use per square or kilo of meat. We then packed and stored the product and carried out analysis during storage to count the surviving Salmonella. And what we found this time, though, was that while the antimicrobial had a small effect, it was negligible compared to the in vitro. So whatever's going on in the actual meat itself was interfering with the effectiveness of the antimicrobial. So the client now had a lot more data to help him decide whether he was going to invest in the spraying equipment or not. So before I finish up then, perhaps a quick mention of just some of the new equipment that the lab is equipped with. I won't go through this whole list, or we'd be here all day. And unfortunately, a lot of the equipment just looks like big boxes on wheels, so they're not very interesting to look at. So otherwise, I would have filled this slide with just pictures. So instead, I'll mention some of the key ones that I think might be of interest. So we have now the ability to do vacuum skin and modified atmosphere packaging in the lab, which is really useful. We have a number of programmable storage chambers that can be used to mimic the temperature changes that are going on during the normal storage and distribution of your product. And some of those will even control humidity levels and light levels as well, which can be very relevant to the shelf life of some products. We have a MinION Genome Sequencer, which is a surprisingly tiny little device that helps us to identify and quantify the DNA and RNA content of bacteria in a food samples. It can give us an assessment of the total microbial community, for example, and also the diversity of that community, so we know what types of bacteria are in the food. And that's a really useful tool, that one. I can see it being especially useful in tracking how that bacterial community changes in, say, for instance, fermented products, stuff like that. We also have a state-of-the-art biofermenter, and that allows us to do benchtop fermentation products. And in fact, this week we have a project starting with kefir, which is a fermented milk product. So I'm going to leave it there, but you can always contact me if you want further details on our equipment portfolio. Thank you very much for your attention, and I'll hand you back to Carol now, and we go through some of your questions.
Thanks, Des. So I'm going to ask Kaye. Yeah, Kaye's there. So we just unmute yourself there, Kaye, as well. Thank you. There's just some contact names. So Des, Kaye Burgess. So Kaye, Senior Research Officer in the Micro Prepared Consumer Food Suite as well. And then our overall Prepared Consumer Food Centre Manager is Shay Hannon. So Shay pulls all of the various strands of us all together, from pilot plant, microbiology, looking at food chemistry. So there's quite a lot of strands to the Prepared Consumer Food Centre. So he tends to pull us all together there. So what I'm going to do is just stop sharing. There's just one, there's a little video that we didn't get to show. But what I'll do is I'll send it out and send a link to that, work something out on that once we have this up on YouTube. So in the next few days, we'll email out to you with where those are on the website. Okay, so we've got some questions here. from, now let me just, sorry. So we had a question here. Does Teagasc have a recommendation for testing water in West Cork? Now, my understanding of that would be a particular testing lab. So maybe we'll ask it from a testing lab point of view first, maybe Des. I'm assuming we don't highlight one compared to the other.
No, I don't. I wouldn't have a particular recommendation. I haven't worked with any labs in Cork for quite a while. But the best thing I would say is to go to the Irish National Accreditation Board website. So that's inab.ie, inab.ie. And they will have a list of all of the accredited labs, and they'll direct you towards the ones that can do water testing.
Yeah, so they...
I think Safe Food well has a list of accredited labs.
Yeah, it's on the Safe Food website. So and as you mentioned earlier, make sure that they're accredited for the particular...
for water, yeah.
Grant. And would you find over the years, Des, accredited labs useful in helping you maybe even design your sampling or your testing?
Yeah, I think most of the good labs will be able to help. It's a matter of talking to them and explaining what you want to do. Yeah, so I've always found them to be very useful, very professional, and yeah, I think they're a good source of information.
So it's about being very clear what you want to achieve and knowing the process. Yeah.
That's it.
Okay, so thank you. And we've got, so I've got one. So how many times should a test be carried out to confirm that the results are meeting specifications? I just ask you that, Des, first. I think that's about repeating.
It depends on the specification. A lot of specifications will give you a number of samples that you have to test. For instance, when you're doing salmonella testing, you usually have to do 5 tests on a particular batch, or 5 batches in a row. So it depends on the specification. Like I think most people, if you're doing routine testing, you just send out a sample. You don't have to keep repeating it, but you'd be doing it regularly over time. So you're looking at a kind of a big picture view of how your sample is doing.
So you're looking at those trends that you were showing us earlier.
Sorry, it's one of those awful it depends answers.
And being really clear what's in the guidance notes and
yeah, you have to look to where your source, what made you decide to do that test? Well, what is the source of that requirement? So on, that's usually what specifies how much testing you have to do.
Okay, and obviously, if you're producing for the multiples, you would have specs there.
Oh yeah.
Okay, so Kaye, just wanted to ask you. There was one on challenge testing, Listeria. So Des talked about the Campylobacter, but do you do challenge testing in the lab now on Listeria?
We can do the challenge testing for Listeria so it's one of the big questions for companies. They want to know, can their product support the growth of Listeria, because depending on whether it can or it can't, that will outline the specifications for it. It is important to note that we're not an accredited lab for doing the challenge testing, but we can do the all of the inoculations and all of the analysis per the EURL spec. But the results would need to be validated by an accredited lab. We have a research project just started working with the horticulture sector, where we hope to do some Listeria challenge testing in this area per the EURL spec.
Okay, and like Des said, it depends. So if someone has an idea for a project, they'd be better to have a chat with yourselves, maybe put something on an email, formulate their thoughts, and take it from there.
Yeah.
Okay, and again, I suppose confidentiality is something that comes up all of the time. So what would you say about that, Des? Maybe if someone's a little bit worried about giving you all the information.
Well, confidentiality is really important to us, and it's been a core part of what we do every day because we're working with companies all the time. I mean, you know yourself, you've seen we have the ability to lock away certain rooms in the process hall, for example. All of our buildings are locked up and secured, and any records that we keep are secured, you know, as per GDPR, which everybody has to work under now. So, I mean, it is something that we value and are really, really careful with, and have always been that way, both with the accredited testing lab when it was around and in our research work. We're always very conscious of it, and I don't think it should be an issue for anyone if they want to come and talk to us.
Yeah, and the more information that they can give you, like that person who, you know, went through the process, and you find out
like
what went wrong.
If somebody comes to me and they want to talk, I will spend about an hour just asking them why and why and why and why, to try and figure out exactly what they want. Because if they're going to pay for service, I want to be 100% sure that they're getting exactly what they want rather than what I think they want. So, but during all of that, that's all 100% confidential, and there's no way we would share that with anyone else.
And Kaye, Des talked about that you could have hosted researchers or someone from your company actually in the labs and working with staff. So I've seen that's a very good. Is that easy to set up?
Yeah, absolutely. We have template agreements in that we would set up with the company. Again, as Des said, there would be an extensive preparatory period where we would talk through with the company what the expectations are on the company side. The staff member that would be hosted would go through the full training in Teagasc to be able to work in our labs according to the SOPs that are in place and following all of the procedures. So everything would be put in place beforehand, and we've always been very happy to work with hosted researchers and have had some very successful collaborations with them.
And speaking of that, COVID at the moment, we're in a level 5, but because we're an essential service, then it's fair to say we're open for business.
Absolutely, it certainly makes life more challenging. But we have robust procedures in place. We have a lot of rotas in place and online booking systems, and we've made a lot more use of online meeting facilities and that, so that where before we may have had face-to-face meetings, anything that can be done online at the moment we will do online, but obviously the lab work can't be done online, and our labs are open for business with enhanced procedures and planning.
Great, yeah, it's just to thank you. And let me just check, we've got another one as well. Okay, so is there, so this is, it's more likely a different department. It's about testing for heavy metals. I presume that's the other side, our chemical side of the.
Yeah, I would say contact Martin Danaher.
Yeah, so I'll answer that. It's directly, and we'll put you in touch with the right person there. Yeah, food safety, just not on the microbiology side. So Des, you would have done training over the years with ourselves. And I'm assuming that option is there potentially if people want some. You talked about people, maybe you're in micro and it wasn't their original knowledge.
Yeah, we've run many times a course called microbiology for non-microbiologists. We used to do it as a 2-day course in Teagasc, but now we prefer to go out to the client's business and maybe do a 1-day course or a half-day course with them, and basically go through, what are microorganisms, bacteria and viruses and yeast and all that. Go through all that detail and then spend a good bit of time on actual setting up testing regimes, sampling plans, and then interpretation of results. So it's quite useful, and we've done it with an awful lot of companies at this point. So it's very good.
Yeah, and
That's still available, yeah.
yeah, we've moved on to virtual training. So we're looking coming up. You can learn how to make chocolate or bone out that that beef, pork or lamb animal, and so we'll be running predominantly online versions of all of our normal courses. And you know, it can be done as a one-to-one over Zoom. Like you said, with a company, but we're making them as practical as possible, so they'll be interspersed with videos and chat. So we're trying to make it as user friendly as possible.
Yeah.
Okay, and you just mentioned a Category 3 lab. We have one on site, Kaye, do we?
I have returned.
Sorry?
I have returned. There was a blip on my internet connection.
Oh, right. Sorry. Okay. So we were, you mentioned a Category 3 lab on site. So what would normally be carried out in that? What type of work?
Okay, so much of the work that gets carried out in that laboratory is on Shiga toxin producing E. coli, or E. coli 057 is one of the Shiga toxin producing E. coli, and they need to be worked with in a more contained facility. So we have a specific closed access laboratory that is to full category 3 standards for work specifically with Shiga toxin producing E. coli and exactly E. coli 057, 026. So within that lab as well, we have the capability to do inoculation studies with these pathogens. For different food types. We also have a range of equipment similar to what's in the PCF lab, you know, so that we can really do the modeling of the food products intended storage profile and packaging regimes and that for these organisms, if that's something that a company is interested in. That's quite a unique facility. Generally, Category 3 laboratories tend to be more in more clinical settings, and that where you don't have the opportunity to work with food products and that. So it's very useful.
Yeah, the whole prepared consumer foods suite, where you can totally mimic the packaging, the shelf life, change the recipes and formulation, and then look at it in real time. There was one that I was looking at. And it's a flow cytometer, Des. Would you mind just telling me what that does? There was one on
I would love to be able to tell you what that does.
I might.
I think Kaye is the expert on that one.
Kaye then, yeah, and then we'll finish up after that.
Okay, so we're interested in using flow cytometry. It's essentially a means to count cells, and they're used quite commonly in more clinical settings for human cells and that, but they can also be used for bacterial cells. So we want to look at the applications of flow cytometry for detecting pathogens in different matrices, but also to be able to look at whether cells are dead or alive in a particular matrix. So you can also use different types of staining in that to be able to look at whether a population is still viable. So it would be useful for examining the impact of different treatments, particularly as we're going for more fresh-like treatments and less harsh treatments. We want to be able to assess how effective these are at reducing microbial numbers. So that is what we hope to do with the flow.
I thought that was a really interesting one. So basically, we're going to finish up now. So if there's some questions that I haven't answered that were outside of the scope, we'll answer those. We'll also follow up with a little email and a links to the YouTube channel and the video. So our webinars continue in 2 weeks. So we've got one of our researchers talking about insect protein and the processes. So it's another one to tune into. And we also have, as I said, so we're going online with our training courses. So thank you very much, Des and Kaye. I presume it's okay for people to email you, seeing you give me your contact.
Absolutely, yeah.
Yeah, great. Okay, thank you very much.
Okay, thanks, Carol.
Bye, thank you.